So in this tutorial we are going to learn about HIV and the effect of HIV on the immune system. HIV stands for Human Immunodeficiency Virus, and it is a type of virus called a retrovirus. So there are three main objectives of this tutorial. The first is to explain how HIV is able to invade and replicate inside T helper cells. The second objective is to describe the consequences of this invasion on our immune system and to describe how this may lead to the symptoms of AIDS. And the final objective is to explain why HIV is difficult to treat, to remove from someone who has been infected with the HIV virus. So first of all, let us look at the actual structure or simplified structure here of the HIV retrovirus. So the first thing to note is the presence of an attachment protein. The attachment protein on the outside of the virus is responsible for specific recognition of the CD4 receptor on the surface of the T helper cells that the virus infects. So because it is specific, it will specifically recognize these CD4 receptors. The other thing to note is the presence of a lipid capsule. Lipid capsule is on the very outside of the virus, in which the attachment proteins are embedded. Next, we have the matrix and the protein capsid. The matrix and the protein capsid are made of proteins, and they are essentially a packaging for the genetic material that lies inside the protein capsid. They do actually have some quite complicated functions, but largely some of these functions are actually unknown, but they are not something that you need to know about. The next thing to note is the presence of the RNA within the protein capsid. So the RNA is the genetic material that the virus enables the virus to replicate once it is injected inside the host cell. Reverse transcriptase is the enzyme that is responsible for converting the RNA into DNA. Once the RNA has been converted into DNA, once it's been injected into the host cell, then another important enzyme called integrase enzyme, shown here, is responsible for integrating the newly formed DNA into the host cell's DNA in the nucleus. So the virus RNA moves into the nucleus of the host cell, and the integrase enzyme causes the DNA to randomly insert inside the host cell DNA. So now we're going to talk about how the HIV virus manages to integrate itself into our own DNA. If a person is infected with HIV, how the virus manages to integrate itself. So, first of all, HIV has to enter our bloodstream somehow, and there are various ways this can happen. Once it's in the bloodstream, it travels through the bloodstream and it binds specifically to the CD4 receptors on the surface of T helper cells that are circulating within the bloodstream. The protein capsid then fuses with the cell membrane, and this means that the RNA and the enzymes inside will then enter the cell itself. Once the enzymes, the reverse transcriptase and the integrase, along with the genetic material, are inserted into the cell, then the genetic material can be inserted. The first step of this is to convert the reverse transcriptase from RNA into DNA. So reverse transcription is normally when DNA is converted into RNA. So reverse transcription is the opposite way around. RNA is converted into DNA. So once RNA is converted into DNA, the DNA moves into the nucleus, and the integrase enzyme is then able to cause the viral DNA to integrate randomly into the host DNA. And this process actually can lead to quite undesirable genetic mutations in the host cell. So, having inserted its DNA into the host cell's DNA, we're now going to talk about how this allows the HIV virus to create new copies of itself within the host cell. So the first thing that happens is the DNA of the virus that's lying within the host cell's DNA. This then is transcribed into messenger RNA, or RNA that then becomes the virus's genetic material. Once the RNA has been created through transcription, this RNA moves into the cytoplasm of the host cell, where the host cell's machinery, so the ribosomes, are responsible for creating the proteins that make up the rest of the HIV virus. So again, it's hijacking the host cell's machinery in order to make copies of all the proteins that they need. Once the proteins have all been made, the HIV virus can be assembled, and there'll be many of these HIV viruses being assembled within the host cell. And then what happens is the HIV moves towards the edge of the host cell, and as it moves out of the host cell, it's going to be surrounding itself with the membrane of the host cell. So this is quite an effective way of hiding from the host's immune system. So how does HIV actually cause AIDS? Because HIV and AIDS are not the same thing. You can be HIV positive, that is, if you have been infected with the HIV virus, you are described as being HIV positive, but you can be HIV positive without having AIDS. And in fact, the HIV virus may lie dormant inside its host for many years before it causes the symptoms of AIDS. So when I mean dormant, that means when the HIV has inserted itself into its host cell's nucleus, and the DNA of the HIV in those T helper cells remains in the DNA of those T helper cells without doing anything else. Only when the host is immunocompromised, or many years later, the HIV starts to make new copies of itself within these T helper cells. So the virus specifically attacks the T helper cells, as we have described before. And because it infects these T helper cells, they are then damaged, dead, destroyed. When the HIV is creating new copies of itself, the T helper cell is not able to do its job. So when someone has AIDS, as in when the virus is actively replicating itself and increasing in number, the AIDS sufferer will then decrease dramatically its number of T helper cells. So it could be by 6 times reduced. And without these T helper cells, then the person with AIDS has their immune system very much compromised, because they depend on their T helper cells in order to activate various other cells needed for an immune response. So the T cytotoxic cells, for example, and the B lymphocytes, as we have studied. Memory cells may also be destroyed. So therefore, AIDS sufferers end up with a very inadequate immune system, and this would leave them open and susceptible to many other infections and also cancers. As we've seen, our immune system is important in recognizing cancerous cells. So, if their immune system is compromised such that they're unable to fight off infections, it will be those infections that will cause the issues that an AIDS sufferer has. They will be a common infection that would easily be fought off in someone with a normal number of T helper cells will become something that's a very serious infection in an AIDS sufferer. So we're going to talk now about why antibiotics are ineffective against treating HIV. So to begin with, antibiotics are generally, they work by damaging the murin wall of a bacterium, or they work to inhibit bacterial enzymes that lead to the formation of this cell wall. So they either directly damage the cell wall or they damage the enzymes that form the cell wall. And without a cell wall, a bacterium is unable to stop the entry of water by osmosis. Usually the murin cell wall is rigid, and just like a plant cell, it will prevent the water entering. Instead, the cell will become turgid. Without the cell wall, there's nothing to stop the entry of this water by osmosis, and that would cause the bacterial cell to burst. And obviously that would kill the bacterium. Viruses do not have these cell walls. Instead, they have a protein coat. They also, it's difficult to target any enzymes that lead to the formation of the virus, because most of the virus's metabolic activity relies on the host cell. One virus enzyme that is often used to try and target HIV is trying to target the reverse transcriptase enzyme. The third thing as to why drugs like antibiotics are ineffective against HIV is that viruses generally cannot be easily accessed because they live inside, most of them live inside the host cells. And even if you kill the ones that are present in the bloodstream, you're still left with the ones that are capable of replicating that are also inside the host cells. There are other reasons why the HIV virus is difficult to treat and remove. So first of all, the HIV is constantly mutating, and this is because the reverse transcriptase enzyme does not do a very good job of copying the virus's RNA into DNA. So mutations are often made in this copying process. So therefore, when the DNA that integrates into the host cell's DNA is then transcribed into RNA again, the RNA will be different from the original RNA. Another reason why it's difficult to target HIV is that the virus is surrounded by a lipid envelope that it has gained from its host. So this is a very effective way of hiding from the host's immune system. And finally, the HIV virus inserts its DNA that's been created into the host's cells. So unless you get rid of every single T helper cell that's present in the immune system, it would be difficult to know you have got rid of every single virus nucleic acid that has been integrated into the host cell's DNA. So as we have studied a few lessons ago, we can use the ELISA test to test for HIV, using a blood sample of someone who we suspect to be infected with HIV. This antibody detection can be used to identify the presence of antigens of the HIV. This method may also be used to test for other infections such as TB and hepatitis, and it may also be used to test for the presence and also quantity of certain drugs in blood sample. So as we talked about a few lessons ago, this is all about detecting certain antigens present in using monoclonal antibodies. So that brings us to the end of this tutorial. So let's think what we should be able to do now. We should be able to explain how HIV is able to invade and replicate inside T helper cells. We should also be able to describe the consequences of this invasion on the immune system and how this invasion may lead to the symptoms of AIDS. We should also be able to explain why, or some of the reasons why, HIV is difficult to treat. So as a final point, if you need to re-listen to any parts of the presentation, simply rewind to the point that you need and listen again, and hopefully if you have the chance to listen to certain parts again, you will be a bit clearer on some of the theory. If you have any questions, please write these down and bring them to the lesson. At least everyone should come up with one question to bring to the lesson. It could be an extension question, or it could be something that you want further clarification on from this tutorial.